Oops. Okay, now. Good morning. There we go. Chapter 18 is what I'm going to go through this morning as far as I can manage if my voice holds out. I'm not sure what's up with that. Okay, so let me go over here. Oops. Here. Okay. Introduction. So, polio. It was once common with potentially serious consequences, including paralysis. So, the iron lung um, was a thing. And there was still someone in an iron lung recently. I should have looked that up. Should have thought of that. We've considered it eliminated in the United States since about 1993. There are still 3 countries in which it's endemic, however. So the overview. So this chapter, we have an overview of specific adaptive immunity, the MHCs and APCs, T lymphocytes and cellular immunity, and B lymphocytes and humoral immunity, and then a brief section on vaccines. Just a basic overview there. So section 1, overview of specific adaptive immunity. So our learning objectives include define memory, primary response, secondary response, and specificity. Distinguish between humoral and cellular immunity. Differentiate between antigens, epitopes, and haptens. Describe the structure and function of antibodies and distinguish between different classes of antibodies. All right, so specific adaptive immunity. Now remember the last chapter we went over non-specific immune responses. These were the inflammation response, responses to pathogen-associated molecules. These are broad general responses, right, that were not specific. When we talk about adaptive immunity, we're talking about specific pathogens. So memory. Like remember CRISPR-Cas9 was one of the systems in E. coli that is kind of its immune memory with the guide RNA that it synthesizes. Well, we have a much more sophisticated system, but that is able to target specific pathogens, and this allows for a rapid response to pathogens to which we've been previously exposed. And it's achieved by programming certain cells. So they have to get exposed first. They're undifferentiated. They're not active. Um, and then initial exposure will trigger a primary response. But then after that, they are primed. They are programmed, allowing for subsequent exposures to result in a secondary response, which is much stronger, as demonstrated here. So if we're looking at the concentration of an antibody that's specific against that particular pathogen, obviously you start out with nothing. And then you slowly develop one that's able to mount a primary immune response. And this, the concentration of this will go away or will decrease somewhat after that initial immune response is over, but it won't go back to zero unless you had measles and it has wiped out your other immunities. This negative immunomodulation, it was a paper in Science. I don't know if I shared that with y'all or not. I can double check after the other business with the take-home exam. Though I'm gonna need to finish that up next. Okay, but so you have a basal amount of this antibody present, circulating, and then if you're exposed to that pathogen a second time, you have a much more rapid and exaggerated immune response that can prevent, expression of the disease. The whole disease stages could be circumvented because your immune system recognizes it and deploys against it too fast for it to be able to establish and cause disease. So hematopoiesis. I didn't, of course, I forgot to fix this figure in the last chapter. I didn't fix it in this figure. That's really annoying. We have our myeloid stem cells and our lymphoid stem cells. From the lymphoid stem cells, we get our natural killer cells and our T, B lymphocytes. Meanwhile, most of these are so for our adaptive immunity, we're looking at the lymphoid stem cells as their source, and the myeloid stem cells for along with red blood cells and platelets We have our mast cells, and then our basophils, neutrophils, eosinophils, monocytes that were all involved in non-specific immune response. All right, so specific adaptive immunity. We're talking about B cells and T cells. B cells mature in the bone marrow. The name comes from bursa, which was discovered in birds, since birds don't have marrow in all their bones. B cells produce antibodies, immunoglobulins. This is humoral immunity. T cells, they mature in the thymus, and these are the central orchestrator of innate and specific cell-mediated immunity, cellular immunity. So humoral immunity is associated with B cells broadly, and then cellular immunity with T cells. So antigens. When we talk about antigens, we're talking about molecules that are similar to the PAMPs in the previous chapter, the pathogen-associated molecular patterns. So those were things like lipopolysaccharide. They were very generic, that were common with a whole bunch of potential pathogens. And when we talk about an antigen, though, we're talking about something that's a unique feature on a specific pathogen. We developed that from, it's a portmanteau of antibody and generator. And it is a molecule that stimulates antibody production and said to be antigenic. So an antigen is antigenic. So in general, more complex the molecule, the more effective they are as an antigen. Obviously, specificity is really high with a really complex molecule, right? Because you don't want cross-reactivity, ideally. So antigens, the anatomy of an antigen, if you will. We have the epitopes, which are the exact little region that interacts with the antibodies, where the antibody directly binds. And then we have haptens. Those are free epitopes that are not associated with a particular antigen. Now, upon association with a larger carrier molecule, the result can be a conjugate antigen. And we do that with certain vaccines, where using the killed pathogen or an attenuated pathogen isn't possible or isn't safe or isn't reasonably safe enough. We can create the haptens and then put that on the carrier molecule to help generate a response. So here's a cartoon for that. This whole thing would be the antigen. These individual little shapes are representative of the epitope. So these are the actual binding sites on the antigen for the antibody, and this gets leveraged for a whole lot of stuff. All right, so the typical protein antigen has multiple epitopes, and that's what we're showing here. There's an antibody that's binding to a couple of different antigens, and this is leveraged in a particular immune response, or that I will tell you about here in a few minutes. So about the antibodies themselves, immunoglobulin. So these are glycoproteins that are present in both blood and in tissue. Okay, hold on. Sorry. There's a dog and a cat getting a little crazy. They are, antibodies. Yeah, the monomer structure of the antibody is 4 protein chains bound down by disulfide bonds, and that's between cysteines. Heavy chain is composed of 2 large identical chains, and the light chains are also identical change. Sorry. Monomer structure, heavy chains, 2 large chains, and then 2 light chains, and they form this white. I'll show you the cartoon next. forming a Y-shaped structure. The monomer it is. And we have the Fab region, which is short for the fragment of antigen binding. The far end is variable, and that's where antigens are able to bind. The epitope specificity of each arm is the same. Okay, here we go. Here's our two heavy chains. They are identical to each other. The two light chains are identical to each other. And here are the antigen binding sites. These are all for the same epitope. Right. Yeah, okay. And then there's the, the wireframe, the whatchamacallit structure. Yeah, and then there's the disulfide bonds pictured there. Yeah, our three-dimensional structure. That's what I'm thinking of. And the FC region, the site of complement and phagocytic cell binding. So that's down here, the part of the constant region. All right, the 5 classes of immunoglobulins. We have our basic monomer, that is immunoglobulin G. We have the IgM pentamer, and the secretory IgA, IgD, and IgE monomers. So, of course, with IgG, IgD, and IgE, those are all, they combine two, they have 2 binding sites. Obviously, you have 10 and you have 4. Okay, wait, those all make sense. The vast majority of circulating antibodies are IgGs, and they can cross the placenta. The rest of them cannot. And they can activate, remember, complement activation. IgG, IgM can do that. And the FC region will bind to phagocytes and IgGs, while the IgE monomer, the FC region, will bind to mast cells and basophils. So here is a bunch of common. So IgDs function B cell receptors as B cell receptors. And then IgEs, activation of basophils and mast cells against parasites and allergens. So remember IgE, I think I mentioned it before. I might not have. You might not have something to remember there. Is involved in the allergic response, as well as response to parasites, to eukaryotic parasites. So secretory IgA is its function. It's good at neutralizing and trapping pathogens in mucus. This is the, again, secretory. This is a secretory component. This is secreted in like breast milk. It does not confer, full. It doesn't. The mother doesn't make like every possible antibody as an IgA and secrete it. These are really effective against gastrointestinal potential pathogens, but it's not blanket immunity reflecting exactly what the mother is able to make for herself. That's what you get through the placenta with IgG. And yeah. All right, so IgG, we have neutralization, agglutination, complement activation, opsonization, antibody-dependent cell-mediated cytotoxicity. And the IgM, we also do some neutralization. Agglutination is a really good, convenient with the IgM. And complement activation. The monomer form serves as the B cell receptor. All right. So neutralization, here's an example. Here's our diphtheria toxin. Remember the AB toxins, the B binds to the cell, induces phagocytosis, and then the A component of the toxin is released. Well, if you have an antibody raised against that toxin, then you can neutralize it. So in this case, we have 2 antibodies bound to the B subunit of the diphtheria toxin, and now it can't adhere to a target cell. Its binding sites are blocked. So IgM, IgG, and IgA have this function. Opsonization is where the antibody serves as an opsonin. So this is able to inhibit infection by tagging the pathogen for destruction by macrophages, dendritic cells, and neutrophils. So these phagocytic cells use the Fc receptors to bind to the Ig opsonized pathogen and initiate the first step of attachment before phagocytosis. So the Fc region, right? So we're binding. The macrophage has cell surface receptors to recognize the antibody, and then whatever it is coded can get destroyed. So it's tagged for destruction. Agglutination is another approach. We saw agglutination before when looking at the virus testing, right? One of the ways you can test for a particular virus. So IgM is particularly good at this. If it's able to bind, so we have 10 binding sites here. If we can bind a few bacteria together in basically a clump, and so they aggregate together, and now they're not able to run around doing pathogen things anymore because they're all clumped up. So of course we have our antibody-dependent cell-mediated cytotoxicity. So antibodies binding to a large pathogenic cell that's too big for phagocytosis are then able to bind to the Fc receptors on the natural killer cells, and so this interaction brings the NK cell into close proximity where it can kill the pathogen through release of extracellular cytotoxins. So there we go. All right, section 2, major histocompatibility complexes and antigen presenting cells. So we have MHC1 and 2 molecules, describe the structure and cellular location of them, identify the cells that are antigen presenting cells, describe the process of antigen processing and presentation with MHC1 and class 2. So the quick, here we are. So the MHC, these are the human leukocyte antigen genes in humans, the HLA genes. I think I might have mentioned this before with the t-shirt sweat test thing, but compatibility, but I'm not going to repeat that now. You can ask me about it on Wednesday. That's 101 for sure, I told. Or wait, was that this? Okay, anyway. MHC, mature erythrocytes are the only cells that don't express MHC. So the language here is that MHC class 1 are expressed on all nucleated cells. And as you know, erythrocytes in mammals are not nucleated. So especially, well, yeah, mammals. So MHC class 1 are the complexes that present self-antigens. So sampling self-proteins, right, and presenting them to the environment to be examined. That's their papers, their passport. Now, if they can't present that anymore, remember the other impacts of that. You can get the release of interferons and whatnot that will suppress the metabolic activities of surrounding cells, maybe even inducing apoptosis in infected cells as a defense mechanism. Meanwhile, MHC class 2 are present on macrophages, dendritic cells, and B cells, and these will present abnormal or non-self-pathogen antigens for initial activation of T cells. So here's their structures. Class 1, both of them have an antigen binding clef. We have this kind of alpha-1, alpha-2 dimer thing happening here with a beta-2 microglobulin. And see it's anchored in the cell membrane. Class 2 is similar in its antigen binding cleft as one would expect, except you have an alpha-1, beta-1 forming the binding region, and then you have a couple of transmembrane domains here. All right, so domains alpha-1, alpha-2, right, and then alpha-1, beta-1 for the MHC class 2. So antigen presentation. So talking about MHC class 2, obviously, and so we have the pathogen that's been opsonized, and it gets, assuming that it doesn't have an escape mechanism, it will be digested by the cell. because you remember lysosome fusion, right? And so the immunodominant epitopes that are associated with MHC class 2 and presented, sorry, I kind of just like how that's phrased, immunodominant, and then are presented on the cell surface. So what does this say? Yeah, lysosome, there we go, proteases, and the most antigenic epitopes, there we go, are selected and presented on the cell surface in conjunction with the MHC class 2 molecules. T cells will recognize the presented antigens and are thus activated. So that's antigen presentation with MHC class 2. Here's antigen presentation with MHC class 1. So again, like I mentioned before and was mentioned in the previous chapter, MHC class 1. Sorry, let me back that up. So infection by an intracellular pathogen can cause an abnormal presentation of MHC1 or no presentation of MHC1, and that will activate immune cells like natural killer cells. So basically you had to have a, I'm okay signal to prevent the activation of the natural killer cell. So cross-presentation, the uptake and presentation of antigens normally presented by MHC2 instead presented by MHC class 1 for CD8 T cells. That's cross-presentation. The mechanism is not fully understood. It appears primarily to be a function of dendritic cells and not macrophages of B cells. I'm missing part of the story here. That's cross-presentation, though. Okay. The uptake and presentation of antigens normally presented by MHC class 2 instead by MHC1 for CD8 T cells. Okay, sorry. That's fine. Not macrophages or B cells. That should be or. All right, section 3, lymphocytes and cellular immunity. Describe the process of T cell maturation and thymic selection. Where are we at on time? Okay, we're good. Explain the genetic events that lead to diversity of T cell receptors. Compare and contrast classes and subtypes of T cells. Explain the mechanism by which superantigens affect unregulated T cell activation. All right. Oh, yay, an SEM that was not colorized. I don't know why that's a pet peeve of me. The colorized SEMs. I think it's just me. I don't know, man. Okay, scanning electron micrograph, right. Spine-like membrane structure increases their surface area, allowing for greater interaction with other cell types and their signals. And as I'm sure you recognize by now, that's a very common theme to get past your volume to surface area challenges. You can have a lower volume but have a greater surface area if you just make a bunch of enfolded areas, right? Like the microvilli. Well, and also like the T cells. Here we go. Okay, so red bone marrow. We have the head of the femur, flat bones of the bodies like the ilium and scapula. So these red bone marrow is the site of production and differentiation of many formed elements of the blood. So our red blood cells, our thrombocytes, or platelets, or leukocytes. Meanwhile, the yellow bone marrow is populated primarily with fat cells. So there's yellow marrow, red marrow. Okay. That's where these originate. But remember the T cells in the thymus, right? So your thymus, or bilobed, H-shaped glandular organ that is located just above the heart. It is surrounded by a fibrous capsule of connective tissues. There we go. Are we looking at some lymph stuff too? Yeah, that's good. Okay, so CD, if you were ever curious, stands for cluster of differentiation. And broadly, for classes of T cells, we have the helper T cells, regulatory, and cytotoxic. So our helper T cells, they have the CD4 surface molecules. These are antigen-presenting cells presenting antigens associated with MHC class 2, and that functions to orchestrate humoral and cellular immunity. It's also involved in the activation of macrophages and natural killer cells. Regulatory T cells, again, MHC class 2 presenting, involved in peripheral tolerance and prevention of autoimmune responses. if things are working like they're supposed to, we're not, well, okay, with autoimmune disorders, which we'll go over Friday, maybe next week, when that's dysregulated, then, we can have humoral and cellular responses to our own stuff. So autoimmune responses. Ideally, normally that's regulated out, and we don't let those get selected for in a healthy, immune system. So cytotoxic T cells, these have CD8 and antigen presenting cells or infected. That's how they're activated. MHC class 2, right. Antigen presenting antigens associated with class 2. Sorry, helper T cells, regulatory T cells, CD4 activated by MHC class 2. And cytotoxic T cells can be activated by APCs or infected or MHC class 1 presenting cells. And of course, its function is to destroy cells infected with intracellular pathogens. T cell, TCR, T cell receptor, spans the cytoplasmic membrane and projects variable binding regions into the extracellular space to bind processed antigens associated with class 1 or class 2, CD4, CD8, CD4. Okay, so activation differentiation of a helper T cell, again, CD4, MHC class 2, and so antigens is extracted from a pathogen, obviously. So we have an unactivated helper T cell or naive cell that is activated by the APC, and then it will proliferate and differentiate into different subtypes, which are type 1, Th1 cells, stimulate cytotoxic T cells and produce memory cytotoxic T cells. Will also stimulate macrophages and neutrophils for more effective intracellular killing of pathogens, stimulate natural killer cells to kill more effectively. Helper 2 cells stimulate B cell activation and differentiation into plasma cells, memory B cells, which we'll go over in a bit, and direct antibody class switching in B cells. Helper 17 cells stimulate immunity to specific infections such as chronic mucocutaneous infections. And memory helper T cells, remember, a specific pathogen and mount a strong rapid secondary response upon re-exposure. Those are our helper T cell subtypes. So the activation differentiation of cytotoxic cells is next. And I'm not going to try to pop that up right now. But let's see. So we have the CD8. Remember the cytotoxic ones. There we go. MHC class 1. This is to induce. So this is obviously the cell itself is infected and is doing an abnormal MHC class 1 presentation. And then preferin, so you're popping holes in the membranes, and granzymes are released. So to induce apoptosis. So superantigens, that was the last little bit there too. So remember, those are from some types of pathogens where we can get and it's got scarlet fever and whatnot. Well, here's how the mechanism works. Normally you have your T cell receptor and your macrophage that's presenting an epitope here. If it's not recognized, you don't get activation of that T cell. However, a superantigen, as in this cartoon, is something that can bridge and force a false association here, and that will activate T cells and cause uncontrolled activation, the release of cytokines, and activating other T cells, or causing excessive inflammation. And that's what will cause what's called a cytokine storm. Now, anti-vaxxers have co-opted that term as well. But anyway, this is how a cytokine storm happens, and that's with a superantigen. So lymphocytes and cellular immunity. That's section 4. B lymphocytes and... Wait, that's... Oh, is that? Yeah. That should be lymphocytes and humoral immunity. All right, production and maturation of B cells. Comparing the structure of B cells, B cell receptors and T cell receptors, T dependent, T independent activation of B cells, and the primary and secondary antibody response, which is slightly more detailed than right at the introduction. Okay, so B cell production and maturation. So B cell receptors are naturally embedded in the membranes of B cells. The available region of all the receptors on a single cell bind the same antigen. So remember we have that, membrane-bound IgD, IgM, and these binding sites here are for the same thing, the same epitope. Negative selection is used to eliminate self-reactive B cells, ideally, right? Immune B cells pass to the spleen for final maturation. Those are naive, mature B cells are passed. And again, the B cell receptors are the membrane-bound IgD, IgM. They will have upwards of 100,000 identical, T cell receptors. It's B cell receptors, up to 100K of them. Okay, so T cell receptors only interact with MHC class 1 and 2. And B cell receptors can interact freely with any antigen or pathogen directly. Obviously, because they have, this is a different, completely different kind of receptor. This is an antibody stuck into the membrane, where the T cell receptors were receptors for the FC region of the antibody. Oh wait, sorry, that's not accurate. They do have that ability too, but we're looking at specifically the MHC 1 and 2 presenting. All right, T cell independent B cell activation can be just by direct interaction. You have a B cell that has these B cell receptors interact with a pathogen and gets activated, and this is creating the pentameric IgM. So the second signal, such as interaction with toll-like receptors and pathogen-associated molecules there are required for activation of B cells. Once activated, the B cell proliferates and differentiates into antibody-secreting plasma cells. Meanwhile, T cell dependent activation of B cells. The B cell recognizes and internalizes an antigen and presents it to a helper T cell that is specific to the same antigen. Here we go, wait. So there we, recognize it there. And we can present it here with MHC class 2, causing cytokine release or whatnot. And that'll induce both the clonal expansion, the memory cells that are not actively secreting, and then the plasma cells, which are actively secreting. And primary response, secondary response, remember IgG versus IgM. IgMs are initial primary response that gets overtaken by IgG, and then secondary exposure, it's the IgG specifically that spikes up really high. And the IgM is about the same, secondary response. All right, so section 5, vaccines. This is a brief section on vaccines. So compare the various kinds of artificial immunity, differentiate between variolation and vaccination, which is fairly easy because we don't variolate anymore. You have to be in a post-apocalyptic world to consider variolation again. And then the different types of vaccines and their respective advantages and disadvantages. Okay, so broadly, the mechanisms of acquisition of immunity, it's either natural or artificial, right? And it's either passive or active. So naturally acquired passive immunity, and that's through the IgA, through the breast milk, or IgG through the placenta. Now, the IgA is not a reason. I don't want to try to get into a breastfeeding rant here at all. Just feed your damn baby. That's all it comes down to in the end. No one should be shamed if you don't produce enough milk, or if you can't for whatever reason. That's fine. You just feed the baby. Formula is fine. Breastfeeding is fine, right? Yeah. Skeptical OB is I forgot her last name, Dr. Amy something. She's a good person to follow on that subject. All right. Passive artificially acquired immunity. That's through serum. So this we harvest antibodies and we give it to somebody. That's one of the responses to. It can be a response to certain emergency infections or highly dangerous infections. That's what antivenin is an example of that, artificial passive immunity. So active, naturally acquired active immunity, it's where you get the illness and then you recover. Presuming you're recovered, hopefully you should have immunity to that pathogen in the future. Now, certain pathogens like I mentioned measles previously will damage immune memory up to 3 years. And the data shows that kids that have measles have an increased mortality to pediatric diseases up to 3 years after recovering from measles. So immune system is damaged and they have to reacquire. It takes about 3 years to reacquire what they had previously. And of course, active artificially acquired is through a vaccine. So I think I just mentioned all this stuff. Oh, wait, antibody transfer, right. Rabies, hepatitis A, B, chickenpox, you can use it for Ebola in 1995. 80% down to 12.5% mortality. And so again, that was serum, right? And then vaccinations. So herd immunity, this is a concept within vaccinations, and this is just basic math. And here's a cartoon. There's a really fun animations for this out there somewhere as well. But if you take your healthy population and you put a couple of people that are contagious in that population where no one's immunized, most people are going to get the disease, right? Obviously. Now, if some of your population is immunized and some people have the disease, well, most people are still going to get that disease. Now, is this showing? No, it's not. It's a little simplified there. That's fine. Sometimes you can be vaccinated and still you can be immunized but not have developed the right antibodies in sufficient quantities, and you can still get the disease later on. Now, if most of the population is immunized and you have a few that aren't because they can't be, there are plenty of medical reasons. Well, there are a few medical reasons that you can't be vaccinated. Then if these people that are not immunized are not exposed, you can't get the disease. So you can never get a disease you're not exposed to. You have to be exposed first. There's a lot of anti-vaxxers that will try to tell you that herd immunity is a myth, but it really is literally basic probability. And I have to work on this because, I don't know, I think it's a pretty good example, but I haven't got a lot of good reaction to it, so I probably need to workshop this. But anyway, this was my example. If you say a vaccine has a 90% efficacy, so 90% of the people that get the vaccine should have developed the appropriate antibodies, against the pathogen. So that means any given individual has a 1 in 10 chance of becoming infected upon exposure, let's say. Now, the exposure means 100% chance of infection in this example, which is kind of true for measles. It's north of 95%. So no exposure, 100% chance not to get a disease. If you're not exposed to the pathogen, you can't get the disease. Now, if you're vaccinated and your friend is vaccinated and they get exposed, your personal risk in that moment is only a 1 in 100. If your friend is not vaccinated, your personal risk is back to 1 in 10. You lose that buffer of your friend. If you go out more degrees of separation, the risk gets even lower and lower. But yeah, basically you manage your own risk by other people also being vaccinated. The best way to manage. And this is if you're immunocompetent and got the vaccine. This isn't even talking about if you're immunocompromised for whatever reason, because your risk of catching the disease then goes up a lot higher. But again, if you don't get exposed, you can't catch the disease. So variolation was originally practiced in the Far East. It spread to Europe and Africa from there. But here's a relief that depicts a patient receiving a smallpox variolation from a physician in the 18th century. But I think it goes back to the 14th century, the practice does. And the idea is that you're able to inoculate a person with a small amount of a pathogen that's healthy, and that person will get enough pathogen to mount an immune response and gain memory B cells from that experience, while not getting the full disease. And even though that does carry risks, and this was Benjamin Franklin, which I don't know, the founding fathers, I don't know, we can't revere them anymore because there's a lot of messed up stuff going on, which I'm not going to talk about here. Okay. Anyway, he wrote in his biography, one of his autobiographies, in 1736, I lost one of my sons, a fine boy of 4 years old, by the smallpox taken in the common way. I long regretted bitterly, and still regret, that I had not given it to him by inoculation. This I mention for the sake of the parents who omit that operation, on the supposition that they should never forgive themselves if a child died under it. My example showing that the regret may be the same either way, and that therefore the safer should be chosen. Variolation for smallpox carried up to a 2% mortality, whereas smallpox I think is up to 30% mortality. Now, modern vaccines aren't that dangerous as that. Variolation was a lot more dangerous because you're giving the wild type pathogen. And so Edward Jenner, he made his initial observations about the milkmaid with cowpox, which looks pretty freaking ugly, but it's a lot less deadly. But you know, infection with that confers resistance to smallpox, right? All right, classes of vaccines. We have our live attenuated vaccines. These are a weakened strain of the pathogen with the goal of establishing a subclinical infection that will activate adaptive immune response. The advantages: stimulates a comprehensive immune response, and possibility of natural transmission is actually an advantage. But the whole shedding thing is bullshit. It is. The low amounts of attenuated virus they found with a couple of attenuated pathogen with a couple of vaccine, or just one mainly, was only found in like the poop, and it was very low. I don't know. Anyway, disadvantages: the long-term storage is challenging, and risks of active infection, particularly for people that are immunocompromised, right? But examples of the live attenuated vaccine include chickenpox, measles, German measles, mumps, tuberculosis, typhoid fever, yellow fever. So again, the disadvantage: difficult to store and transport, risk of infection in immunocompromised patients, but it does confer cellular and humoral immunity, and it is long-lasting. Inactive vaccines, that's where we use the whole pathogen that has been killed. It's easier to store and transport, no risk of active infection. However, it also gives a weaker immune response, requires higher doses, and requires boosters. Subunit vaccines, these are where we only take the key antigens and that we produce by genetic engineering. The toxoid vaccines are the inactive bacterial toxins. Conjugate vaccines, protein conjugated to a capsule polysaccharide to stimulate the production of antibodies against capsules. All right. Oh, and that was left over from the risk of reversion, transmission to contacts from the, there we go, attenuated. So inactivated vaccines include cholera, Hep A, influenza, plague, rabies. So these are killed, heat, chemical radiation, whatever. No risk of severe infection, but does require boosters. There's a clinical trial starting for the universal flu vaccine now. If that turns out well, we will have a vaccine. You only have to get it once. Well, no, it might need boosters, but I'm not sure. But you wouldn't have to get it annually, is the point. Okay, subunit vaccines. So these are just the antigens, lower risk of side effects. But the disadvantage of limited longevity, multiple doses required, no protection against antigenic variation. This is our current problem with pertussis, whooping cough. We lost at least one of the antigens that we were using in the subunit vaccine. That's the acellular, the Tdap, or the DTaP. AP is acellular pertussis. But this also includes anthrax, what most people don't get, Hep B, influenza, meningitis, the wart virus, HPV virus vaccine, right? Pneumococcal pneumonia, toxoid, botulism, diphtheria, pertussis, tetanus. So the T is a toxoid in the Tdap, in the D. It does not prevent infection, however, but it will neutralize toxins. And the conjugate vaccines, So again, this is capsule polysaccharide conjugated to a protein to give a T-dependent response to capsules. A better response in young children, but it's costly to produce. No protection against antigenic variation, may interfere with other vaccines. That's meningitis. And the Hib, the strep pneumonia, Neisseria meningitidis. And of course, anti-vaxxers. I'm not going to go on long because we're already almost at 50 minutes now. But they've been around as long as there's been vaccines, variolation. It doesn't matter. They've always been around. They will always be around. And that's a different subject, ultimately. But anyway, that brings us to the end of this chapter. I'm going to go off in a corner and do a bunch of coughing, I think, next. And I guess catch lunch with Nick. All right. Anyway, have a good rest of your day. And I am out of here. Oops. As soon as I can do this. I'm not doing this cleanly.